whilst we're preparing for it, thank you very much for the introduction. As you heard, it's interactive, so I will give you a presentation, and in between there will be questions, quiz, but also opinion polls. Sometimes it's difficult to distinguish. And what we're just doing, we're distributing cards, 3 colors. That's the old analogous Kahoot system. And as soon as the cards are distributed, I will start with a question just to make sure that you understand what we are doing with the cards. So everyone receives 1 blue, 1 red, and 1 yellow. And there will be usually 3 questions, questions. And if you think you can identify yourself with the one, or you think that's the right answer, you just put up the card, the color, which is shown here then in the question from the... Sorry?
Anybody from the faculty also wants to play?
Yeah. That's important to see whether they understand it. Okay, ready? Okay. Okay, we start with the first question. The first question is just to make sure that you can understand the system. So, which is the right answer? Up, up, up. 100% you all understood the system. Okay. And the next few ones will just do questions to warm up, but it will get more and more difficult at the end, I promise you. Yeah. With what can you especially please the molecular diagnostic lab? With empty request forms, unlabeled material, or properly filled in request forms with clear instructions? I'm surprised you all know the yellow, because that's not usually the case. At least 10 to 15% of what we receive are the empty request forms or unlabeled material. And the causes, of course, a big problem for the lab, because usually you should refuse and reject the material and go back, and then you have to repuncture the patient, bone marrow, something like that, or you make a biopsy. So that's a very difficult issue and also legal issue. So I urge you and your colleagues always to fill up the request forms and communicate with the lab and tell them with the information or provide them with the information they need and give them clear instructions, which is, of course, for you sometimes very difficult because you don't know very much about the technology and methodology. So there are clinical considerations for the molecular genetic diagnosis. You for yourself have to define the diagnostic or research questions. You have also to decide which type of material is adequate. So, I mean, if you have a leukemia, blood, bone marrow, it's more difficult with solid tumors because usually it's very difficult to get fresh tumors. You can get fresh frozen material, and formalin-fixed tumors is not necessarily always the right thing for what you want to do and what you want to analyze. And then, of course, you should also provide the lab with the information what you're looking for. What's more important is the gene fusions, copy number abnormalities, mutations, expression patterns. Is it the whole genome you are interested in, or only such specific regions? What do you want to know? Do you know genomic lesions, or do you want to know something about the expression patterns, which is, for instance, interesting if you look for targets for signal transduction pathways? And also, if you do molecular sequencing, for instance, it's very important also to define the sensitivity which you require. A diagnosis is probably not such a problem, but in the follow-up samples, it's more important to have a higher sensitivity. So here's the next question, or actually opinion poll. What is for you the most important motivation for requesting molecular analysis? Is it the prognostic stratification, which is just required by treatment studies? Are you interested to screen for specific treatment targets, for instance, in resistant cases or in relapsed cases? Or do you want more to obtain a general genomic overview that may disclose novel findings of potential importance? So you have to decide. Okay, it's not always the same color. The first part is, so to say, very easy to fulfill, but not very rewarding, I would say. Yeah? Because you just get information which you need for the treatment, but besides that and above that, you don't get any new information and nothing for the future. Screening for specific treatment targets is important in cases which relapse or which are treatment resistant, but I, for myself, and always propagated, you should do for a diagnostic strategy the best you can do to get everything what you can get above what you just need for the treatment study, because it's a minor issue. I know instances that, for example, something was ignored in their last treatment study, and for the new treatment study, you would need it for stratification. And then you have a problem because you don't have the information, not the data. And there are many tools, of course, which you can use for the genetic workup of malignancies. There's the old-fashioned ones, which I grew up with and I'm very well acquainted with, which is cytogenetics and FISH. There are the molecular genetic methods, which are basically PCR, also expression analysis and looking for fusion transcripts. So that's basically the old ones. And these are the new ones. That's basically whole genome sequencing, DNA-based, and that's what I mainly will talk about now. What's the significance and what you can do with it. And to just get an overview about what's available here is that if you just look at DNA, you can do targeted sequencing only for specific regions, whole exome sequencing, which is the more common part now. It's cheaper than targeted sequencing, and from the whole exome sequencing, you can actually virtually retrieve targeted sequencing, targeted genes. So it's cheaper to do whole exome and just extract which genes you want. And whole genome sequencing, which is now on the uprise because it's getting cheaper. On the RNA part, you have, so to say, the whole transcriptome sequencing for gene expression analysis, or you can also have targeted sequencing, for instance, just looking for fusion sequences, but fusion genes, but you can also get that, of course, from whole transcriptome sequencing. So the three most relevant sequencing approaches are the targeted sequencing, whole exome and whole genome, as I told you. They have different applications, different advantages, different disadvantages. Targeted sequencing was very in favor before whole genome sequencing became cheap, before whole exome sequencing became cheap. And you get the sequencing depth of 500, and it includes all the abnormalities which you find in the genes which are in the target panel. Whole exome sequencing, as I said, is now the most common method to do, and you sequence the whole exome with a sequencing depth of 50 to 100 fold, and it also identifies all kinds of abnormalities, and it's at the moment more cost effective than targeted sequencing. Whole genome sequencing, you sequence basically the region, the whole genome, although whole genome does not really exist yet with sequencing, does not cover the whole genome, you have to be aware of that. And with a sequencing depth of at least 30 times, and it covers everything, also, so to say, all parts in the genome which are not exomic. So here's the next question. What do you think? Whole exome covers approximately 1% of the entire genome, 5% of the entire genome, or 10% of the entire genome? That's a bit mixed now. It's about 50-50, I would say, between red and blue, and a few yellow. So actually, it only covers about 1.1%. So if you do whole exome sequencing, you must be aware that's only a very, very small part. And besides that, different exome kits which you use cover different regions, and not all regions about the exome. So you have the genome, exome, and then the transcriptome. And about 80% of the exons are less than 200 pairs long, base pair longs. That's why it is very good for exome sequencing, because it's short read sequencing. And it constitutes about 1.1%. And only about 30 megabases of DNA from about 3 or 6 billion base pairs in the diploid genome. But on the other hand, it seems to harbor 85% of mutations. But I think that's a wrong estimate, because if you just look for something under the light, you only find what's... If you lose your keys somewhere and you look under the light, you don't find the keys. So, because you do only exome sequencing, it's a very high percentage, but if you would do whole genome sequencing, it's much less than in the exome. There are short and long read sequencing. I don't want to go into details yet here too much, but Illumina sequencing, which is the most common one, is a short read sequencing. That's why it's quite good for exome sequencing. And there are two new long read sequencing methods. One is bulk biosequencing, which is very elaborate, extremely expensive, therefore not appropriate for diagnostic purposes. And nanopore sequencing, which I will show you how that works in the next slide. Oops, sorry. Nanopore sequencing is a very interesting method because...
Nanopore sequencing relies on the use of...
Sorry. Was pre-recorded before. So you only have something which is a USB stick which you put onto your computer, put your isolated DNA there, and this stick here has many such nanopores here. And these nanopores are protein pores, and through electricity or with electric current, you feed through single-strand DNA here. And because every base pair has a different current, you can actually deduce different resistance. You can actually sequence that. And you can sequence the whole genome with that, long read sequencing. And it's very convenient and not too expensive. So it's a very, very nice method, especially if you only have a low throughput, only single cases. Another method, which is not really a sequencing-based method, but it's very fashionable now and becoming very fashionable, is optical genome mapping. It is supposed to replace old-fashioned cytogenetics. And the principle of that is basically gel electrophoresis of single-strand DNA. So you need a high molecular weight DNA. You label that with a specific enzyme and transfer the labeled DNA to a cartridge, and then do an electrophoresis. There are many nanopores also, or channels. And through the channels, you feed through the electrophoresis single-stranded DNA. And because you have labeled them with an enzyme where you know where the enzyme is located in the sequence, the system takes many pictures, and from the pictures and the distribution of the label, it assembles the whole genome, and then you get such a pattern. And from this pattern, it's very easy so to say, to define everything from translocations, deletions, duplications, something like that. Overall, very, very good, but also has its problems, and that's become very fashionable nowadays. It's also not very cheap. So something which is dear to my heart is the problem between mutation and SNP. So what do you think is the difference between a mutation and a SNP? Is no difference? Is it just different names for the same thing? Or as the general knowledge knows, mutations are pathogenic and SNPs are benign? Or is it a difference in the frequency, population frequency? So please vote. Not everyone dares. Red? Yellow? Are there any blues? So red and yellow are principle right. Yeah? So there's no difference. It's the same thing, but it depends on the frequency in the population. So everything which is lower than 5% in a population is called a SNP, and everything which is more than 5% is a mutation, which has its own problems because it depends on the ethnicity. If you look, for instance, for factor V Leiden, in Japan it's very rare, so it's a SNP, and here it's more common, so here it's a mutation. Also lactose intolerance and whatever. So it's not a very good distinction, but it's basically what it is. So please be aware and tell your colleagues the mutation is nothing bad, and SNP is not always something good. That's the message. And in the genome, we have more than about 12 million SNPs, many more now, because they have now sequenced all the populations and put them together, which are approximately 0.4% of the whole genome. And you have it distributed all over the genes in the exons, but also most of them are also in the introns located. And there are thousands of SNPs per chromosome, and you can get SNP profiles. And if you do whole genome sequencing or arrays for SNP arrays, then you can determine the different patterns of the SNPs. And that is useful, for instance, also for population screening or for identifying specific predisposition regions, not necessarily mutations, but regions of genes, or also in introns, which are associated with a predisposition to particular diseases. So what you do here, you have a disease, control and cases, do genotyping, get SNP patterns, or you have unselected samples, and then you get something like this plot here, and whatever is significance here is a region where it's important. So for instance, in leukemia, I think they're the best, as far as I know, the best known examples were looked at, and especially in ALL. There are quite a few such SNPs known, which not really predisposing, but they have some predisposition. They are very common in the population, yeah, but enriched also in the leukemic cases. And most of, or basically all of the SNPs, which we know now in leukemia, they're not located in exons, but outside in the introns, and affect probably also more or less regulatory elements of particular genes. So it's a regulatory difference and not necessarily something which is directly located in the genes. And this refers to normal genetics, but it's also true, of course, for cancer tissues, that you have different types of genetic variants, high ones with high penetrant mutations, which are the Mendelian disorders, rare variants with small effects, and common variants with small effects, and all the different types of mixture. So you need a particular pattern, usually, not only a single gene which makes an effect. And that's important also to know that if we have also talking again about SNP and mutation, that nothing is really black and white, but you have many, at least 50 shades of grays in there, which have to be in a combination and increase or decrease the likelihood that you actually get either a tumor or leukemia, or within the tumor or leukemia, that it gets more aggressive or less aggressive. And to come back again to the relationship between mutation and the SNP, as we said before, and as you're all aware, the terms mutation and polymorphism often leads to confusion in the communication because of incorrect assumptions of pathogenic and benign effects. And now there's a general agreement that both terms should be replaced by the term variant. It's a neutral term. With the following modifiers, you can have a pathogenic variant, likely pathogenic variant, variant of uncertain significance, or likely benign or benign. And there are many SNP databases, or big SNP databases, where they collect all the also disease-associated SNPs, where you can actually look it up and have algorithms and programs where you can determine basically how likely it is that it's somehow connected with a disease or responsible for a disease. So we come now to acquired mutations. Another question: are acquired mutations restricted, are always restricted and specific for the tumor tissue? Can they also be present in several normal tissues, in a tumor patient we're talking now about? Or can they also be present in all tissues? What's your opinion? Yeah. Yellow, blue. Why not all three? No? Yes and no. I'll show you. Actually, they can either be restricted, they can also be present in several normal tissues, but they can also be present in all tissues. Depends. Huh? But you only had two. You can distinguish between germline, de novo, and somatic sequence variants, where de novo and somatic can be more or less the same thing. So if something is inherited, you have you have a father here, which is a mutation carrier in all his cells, and of course the germ cells are also. So his son or his son or daughter, they're always heterozygous carriers. If father can be a mosaic but affected with germ cells, then of course it's the same situation here. All cells in the progeny would be mutated or carry the mutation, and you can have only germ cell mosaicism, which is, for instance, the case quite often the case in RAS mutated cases, where you have germline mutations already in the germline of the father, but you can really detect it. And then, of course, the patient has also a mutation. You can also have a de novo mutation, so the parents are not affected, but the mutation occurs in the child. And it depends on when the mutation occurs. If it occurs already in the zygote, then you have affected all the tissues. But you can also have, so to say, tumor-specific mutations, where somatic mutations are only in the tumor and not in other tissues. So here the distribution can be, patient can be a mosaic in different tissues for mutated or non-mutated tissues. So the important thing is to delineate and acquired from pre-existent germline sequence variants in tumor tissues. Usually start with the tumor tissues, you look at the tumor tissues, and then you get sometimes into trouble because you find something which could also be constitutional. And then you have to basically look what you do in order to identify that. So the most important thing for clinicians is also to look at clinical stigmata, because there are many syndromes which make tumor prone. You look at the personal family cancer history. You look whether there are any signs of cancer, high microsatellite instability. If you find mutations in a gene, it's you have to check whether it's a cancer susceptibility gene. You have to distinguish between, as I said before, variant pathogenic or likely pathogenic variants from benign ones. And you can also look at the allele frequency. If you do sequencing, you have 2 alleles. And if you have less than 50%, it's very unlikely that it's a germline variant. Because at least if you have a heterozygous carrier, it should have at least 50% of the tissue or of the DNA mutated, because it's a heterozygous. And if you have more than 50%, it could be also an additional homozygous mutation than in the tumor. So 50% is more or less the cutoff where I can say yes or no. Or you have multiple primary tumors in the same variant. You can check for the ethnicity of the patient. In some ethnicities, Jews for instance, there are founder mutations, which are quite common and usually always the same type of mutation. Or patients with pathogenic cancer susceptibility variants, which if we identify such cases, you definitely have to do germline testing, but before you can do that, you have to look at all the ethnic and data protection issues and refer them to genetic counseling. So we come now to the RNA, whole transcriptome sequencing, which is basically gene expression profiles. The most important 2 things are looking for fusion genes in there, and also to look at the gene expression profile for signal transduction pathways. The problem it seems very easy to say with RNA sequencing, you can look for fusion genes, and but if it depends very much, because first of all, many of these fusion genes are very low expressed, so it's very difficult sometimes to find them. And then it very much depends on which algorithm you use, whether you identify proper fusion genes or not. You have here 1, 2, 4, 5 different algorithms, and the number of fusion genes to detect. And if you use all 5 algorithms, you only find 14 which are the same fusion genes. So the difficulty here is basically the analysis and the bioinformatic analysis. So no program showed satisfactory performance. The performance varies very dramatically in terms of precision, sensitivity, and computational costs, and according to mapping methods, filter strategies, and parameter optimizations. Programs with a high sensitivity predict thousands of false positives. And programs with low sensitivity still produce tens of hundreds of false positives, an unacceptable number for experimental confirmation, and with very limited overlap in the results. Of course, if you deal with such issues very often, then you get some feelings, or if you know which tumors you're looking at or which leukemias you look at it, you get a feeling what you look for and what's more likely and what's less likely. And you get also, of course, a list which is listed according to the likelihood that it's there or not. But it's not such an easy issue as one would imagine. And in between, there's another question. Gene fusions are highly specific markers for which forms of solid tumors? Sarcomas, neuroblastomas, or osteosarcomas? Anyone without red? No? Very good. So the purpose now, or the aim of all this genomic sequencing is, so to say, to combine everything in the multi-omics premise and promise. So to identify methods which one size fits all. There are programs now like Inform where they do everything, all the different methods, try to combine them with algorithms or something like that. But as far as I know, they were not that successful in really helping to identify specific targets or pathways which can be treated. So it's not a very good approach yet. But nevertheless, that's what we look for and what we aim for. And I will talk now to you something about the future developments in molecular genetic diagnostics. And the good news is that the paradigm is shifting. And during my 40 years in genetic diagnosis, the paradigm has shifted many times already, starting from cytogenetics, FISH, PCR, sequencing, arrays, or something like that. But this, I think, is really kind of very revolutionary and very, we can expect quite a few things from that. And before I do that, I have another question. Is it possible to obtain a complete overview about the genomic and epigenomic structure, as well as the expression profile of a tumor without ever even having access to the tumor tissue itself? I hope you understand the question. You do molecular genetics, but you have no tumor. Most of them, not everyone has decided yet. Yes, very optimistic. And perhaps you're right. And the way to go is cell-free tumor DNA in the plasma, or actually, the whole story started with prenatal diagnosis, where they identified cell-free DNA in the peripheral blood of the mother, where it could actually do non-invasive diagnostics for the fetus or fetal abnormalities like Down syndrome and all the mutations now and everything else. And the thing is that here, example for lung cancer, because an adult patient, paper about an adult patient. So you have different things. You have also tumor cells in the peripheral blood. You can have necrosis and apoptosis. And by that, always DNA is set free, also RNA to a certain extent. And then you can, if you do sequencing of the cell-free DNA, identify point mutations, copy number operations, rearrangements, also methylation changes, which are the most important things now. And as you probably will hear tomorrow by in the predisposition about ZNS tumors, also mutation signatures. And the principle is now, if we look at one cell here, then average yield of a nucleated cell is 6.6 picogram of DNA, which is considered one genomic entity. And if you have an average of 1 milliliter of plasma, you get already about 1,500 genomic entities on average. In a normal, even a normal patient, not a tumor patient. But if you want to look at the tumor, you can determine or distinguish the tumor fractions in a patient with a tumor from the normal free DNA. And that depends very much, so to say, on patient sample, how big the tumor fraction is. The tumor fraction is not always aligned with the tumor size. So sometimes they have big tumors and less, or small tumors with more. And then the analytical limit, of course, of detection of genome-wide targeted sequencing, where you have the genomic coverage and the analytical sensitivity, which can be low or high. But the whole thing, the beauty is that if you look here, you have cell-free DNA, and you have a specific bioinformatic algorithm. And if you have a tumor fraction which is more than 10% of the cell-free DNA, Yeah, you can compare that, and you can also separate the cell tumor fraction from the normal DNA. And what is shown here is the different approaches, but the most important thing here is that if you have a tumor fraction of 0.27% in the peripheral blood, you only need one amplification. So in principle, that's a cheap method compared to if you do the whole genome sequencing of the tumor, you do 50, 100,000 fold. Yeah? So one fold, and you get a nice pattern of copy number aberrations, but also you can identify mutations and something like that. And here's one practical example, which was published about 2 years ago in the in Blood. Was about Hodgkin lymphoma. And the interesting thing is, of course, you only have Hodgkin cells which are abnormal, so there are very few, in principle, in a Hodgkin lymph node. And still, you can, on a cell-free DNA, you can identify copy number aberrations, which is virtually impossible to do so far on, I don't know, you would have to separate the cells. And if you know these abnormalities, of course, you can define fish markers where you can look at the tumor cell or smear or slice of the tumor, and you can verify whether that's correct or not. But you can also identify different types of mutations, also the fraction of the mutations here, and that you can use for follow-up investigations before therapy, 1 cycle and 2 cycle, and you can do in principle that as often as you want because you have access to blood. Depends on your costs and your interests and what you want to do with it. But in principle, it's a nice, so to say, follow-up method for that. If you identify mutations which you can follow up. And there was a recent paper also from research institute in Vienna about Ewing sarcomas, where they looked at the number of Ewing sarcomas. And what you can also see here, it's not only DNA which you can identify, but you can distinguish the tumor DNA from normal DNA, but you can also, depending on whether the chromatin is open or not, and whether during apoptosis that's cut or not, you can identify basically whether it comes from the tumor or from other tissues. And here you see that the difference between normal and tumor tissue in the DNA is usually, the fragments are much smaller. And you can do a lot of things with that. You look at the chromatin, you can look at the methylation, you can look at the mutations, something like that. And the cute thing is also that you can also not only determine from which tissue this DNA comes, according to its methylation, basically, or you can do RNA sequences. Not only the RNA is not very stable in there, but nucleosome occupancy maps, where you can identify whether open or closed segments, which genes are expressed or not, and also methylation maps. If you put that together, you can do the tissue deconvolution, and you also can identify so which tissue contributed how much to this whole thing. So and I think I'm not quite sure, but yes, I'm in the end now. And now comes the most critical question for me, but also for you. Thank you very much for attention. Red or blue? Unfortunately, you have to show me red now because it would be for both of us very embarrassing. Thank you.
Are there any questions?
First of all, Oskar, very inspiring. I would like to be 30 years younger so I can see the end of this, the future is already here. No? But basically, based on the things you presented, you can diagnose a cancer before it is clinically evident. At that point, you can also follow the disease as minimal, residual, or disseminated. So my question is, from the samples, are there additional factors that can change the results of the samples? Day, night, run, rest, male, female? No, I don't know. It's just a question.
No one has looked at that.
Okay.
But I think you need first the clinical suspicion before you can identify the tumor, because it's not a screening method. But what you can do, for instance, if you have problems with a solid tumor and you don't know, I don't know whether it's a Wilms tumor, neuroblastoma, or what it is, or even if you find an incidental loma, yeah, you could in principle identify which type of tumor it is through this method. Yeah? And the one thing which I forgot, but which I found is also very interesting, is, of course, if you do it on a large scale, and I don't know, 100 samples at the same time, it's very cheap. Yeah, it's also, compared to a whole genome sequencing of the tumor, very cheap. But in principle, you can also do it on a MiSeq for a single case. Yeah? So it's more expensive then, but you can do it. Yeah.
Well, I was just thinking, I don't know whether you can do it from blood spots, but we've got, of course, the neonates who give their blood spots, and we've got several tumors presenting early in life. It would be interesting to see if you could find early findings. But I don't know if you, within blood spots on paper, for the screening.
No, that has been in principle done, yeah, but on cells, yeah. I don't know. You cannot distinguish plasma, dried plasma in cells on this blood spots. Yeah. Yeah.
We have a question from the audience, but I also wonder, who remembers we had a very interesting Time magazine article on this? Who wrote it again? Yes, it was you, right? It was on
What?
screening in blood for tumors. You were the future, yeah. This is old news. We already learned this from one of the participants.
Unfortunately, I only read scientific literature and not Times.
Thank you. That was a really interesting talk. Maybe a controversial question, but does this herald a future where we can diagnose all tumours from a blood test? And what does that mean for research on tumour tissue going forwards?
So I think it will be a bit similar to prenatal diagnosis, yeah? Because you don't have to access the fetus, or in this case the tumor anymore. But you could do it, you can basically do most of the diagnosis, and many of those things you can't really even do in the tumor. I mean, the advantage or disadvantage is of course, you don't know which parts of the tumor is. It's a general overview, yeah? But of the main clones, basically, and of course in the tumors you have different batches of different abnormalities and so on. But even then, if you do just sequencing or analytical methods on the tumor, you're never quite sure whether you can see all the patterns. Yeah? That would be a method for single cell sequencing, but even then you only look at, I don't know, about 1,000 cells or something like that. So it's also not the conclusion. But here, I think you can at least get a very good overview. I mean, if you have enough tumor DNA, that's also the question. Yeah? But you get a very nice overview, general overview about what's happening in the tumors. And what's not quite clear yet is where this DNA comes from. I mean, it comes from apoptotic cells in principle, but what causes the apoptosis in there is not quite clear. Yeah? But in general, I think it's one way forward and a big step in that.
So I had a question, but you have already partially answered. So just asking, what do you think about single-cell sequencing? Do you think it's going to be easily translate to clinical practice, or we are so far at the moment?
As a clinical diagnostic tool? No.
Yeah, in the future.
No. I mean, it's a very, very important, very neat experimental tool, and you can get a lot of interesting questions and answers for that, yeah. But for diagnostic tools, I think it's also out of scope because you can only manage a few hundred cells or something like that. And it's also a very, very, so to say, elaborate method, so, and also not cheap. Yeah. I mean, that, for instance, I think the problem here is, for me, it's out of question because I'm used to cytogenetics in fish, yeah. But my problem also is everyone forgets about the old methods and starts only with the new methods. Bioinformaticians don't know anything about the rest, yeah. So it would be very nice to integrate both, the knowledge of both, not necessarily method, but the knowledge. And you also have to be careful which bioinformatic tools you use in there. But in principle, I think that's one way, the only way forward nowadays. And it's comparatively cheap once you have the algorithm, bioinformatic algorithm set up. From a methodological point of view, it's not that difficult, I think.
Thank you for a really interesting presentation. This is going to all these kind of techniques are going to generate more and more data about people that is going to need to be stored somewhere and, you know, potentially accessed in the future. How do you think this kind of information from a cancer diagnostics point of view, maybe in a very young child, how should that be used in the long term? Who should be able to access that? Just the patient, companies, insurance companies, health services? Be interested on any thoughts you might have about that.
I think, as I heard, that's the topic of the next module, ethics, data protection, and something like that. Yeah. But in terms of storage, if you just do, so to say, one-time amplification, the amount of data are not that big. If you do sequencing of tumor tissues with 100,000-fold, yeah, that's enormous amount of data. And there are always still questions, what should you do? Should you just use or collect or store the processed data or the raw data on the one hand, yeah? And there were times, and still it is now the same thing, that rather than storing the data, you resequence again because it's cheaper.